Hello, everyone. Good afternoon. It's a privilege to follow Mauro's talk. They're going to be talking. I don't know where you went. Wherever you are. Oh, hi. There you go. Great job. So there's a lot of relevance between what he just presented in terms of his rebound in a dish and what we're trying to do with the sort of more direct analyses of the samples from this trial and looking at rebound in real life. And I think this allows for multiple things, but a comparison of this model of rebound in a dish with rebound in real life, but thinking about how multifactorial any biological processes, but particularly time to rebound or suppression after an intervention, there's so many factors that go into the reactivity of the reservoir, the level of latency, the host immune responses, the intervention. And so we're trying, I think, within this collaboratory to put many different investigators together with one group of one trial, one cohort, one group of samples, and hopefully get into some of the complicated and interweaving relationships. And so I think this B2006 participant has sort of risen to the top of Matthias and Maxime's work and Mauro and Joe and the Silicano's work, and certainly when we look at these samples. So it's fun to see the connectivity. So I think for the work that we're doing, trying to assess these rebound samples and then working with Matthias and Maxime to look at the reservoir viruses, our goal is to think about this question that Mauro brought up, that there is a disconnect between the sampled reservoir and the viruses that reactivate. And how can we dig into that and understand that better, explain the results of the study, and then also think about sort of the key features of our immunotherapy. So if we start from this study, this BEAT2 study, which I know we're all familiar with, but again, their participants were given maintain on antiretrovirals. These are chronic ART initiating participants. Maintain on antiretrovirals given a period of time where they got type 1 interferons, then added on 2 bNAbs, 3BNC, and 101074. The antiretrovirals were stopped at that period of time. That continued until the sort of primary outcome, this time point right here. And then, in theory, the antibody levels waned, and they waited for treatment for virus to reactivate. And so the question that we wanted to do, starting with characterizing rebound viruses and their sequence characteristics, phylogenetic relationships, and phenotypes, is ask how did those administered broadly neutralizing antibodies, as well as the autologous antibodies that are shifting and evolving based on whatever antigen they're exposed to, how did they contribute to virus suppression or selection by looking at the differential pressure against reservoir viruses and rebound viruses? And so in a very brief conclusions for sort of everyone, the community and all of us, we found in very broad strokes that bNAbs can durably suppress viremia. That's the sort of primary outcome of this study. But resistance in the reservoir and the rebound are associated with those early breakthroughs. We also see in some participants that autologous neutralizing antibodies may contribute to some of the suppressive pressure in this study. And so these data, I want to emphasize kind of two things. These data are new. They've just sort of come in this week, and so there's a little bit lack of synthesis on my part, and I'd appreciate anyone's thought, but I wanted to get the chance to talk about it with this group because it is a sort of multidisciplinary approach. But also, it's also very much dependent on the collaboration of all these different groups, from the study team to Matthias and to the Silicano. So hopefully we can all put our heads together. So again, the overall clinical outcome of this study was that during the 2 bNAbs and the PEG interferon treatment period, there was good suppression for all of the participants except 2 who experienced virus rebound. And then as those antibodies waned, there was continued rebound for many of those participants. So here are the viral curves from the 2, or viral rebound curves from the 2 individuals who broke through high antibody concentrations in step 3, whereas the rest of the participants either rebounded as antibodies waned in step 4, or, sorry, I can do this. Or we had these sort of participants who had prolonged times of virus suppression, including some that we consider having a post-treatment control or post-intervention control phenotype. So I'm going to walk you through the analysis we did for a few of these participants just to get a sense of what we did and then hopefully come up with some general themes. So if we look at participant 11, and this is the viral rebound curve in these purple squares, we took these initial rebound viruses, first detectable virus, and then subsequently for every detectable plasma virus we had on a week-long basis going through. So this is the OM sequences from plasma virus. And we're starting here at the first detectable viremia at 43 days post-rebound, and we're going for about 50 days through 92 days. And what you can see is that this is our scale. We have a very homogeneous lineage. So our first detectable rebound are nearly identical. We see minimal virus evolution over time. And in fact, over these many weeks of viremia, we actually see only a few amino acids of novel mutations arising in this group. So this is a single lineage that's adapting with potentially good or important phenotypic consequences, but not a lot of diversity and no new lineages arising. So we cloned one of the original viruses shown there, as well as one that was sampled at the latest time point that's actually only a few amino acid differences, and we asked, what's the sensitivity first to the administered broadly neutralizing antibodies? So what we saw in this individual was frank resistance to 101074 and IC50 greater than 10 micrograms per mil, and sort of moderate sensitivity to 3BNC117. And so if we put all of the participants together and we think about those early rebounders versus those that rebounded during step 4 as the antibodies were waning, and those that had much later rebound, we can see there's this nice relationship between early rebound having the most resistant viruses, in particular to 1074. Similarly, in our mid-level rebound, quite a bit of resistance. And unfortunately, we were unable to sample virus from one of the late rebounders, but you can see there's sort of a trend towards greater sensitivity in those individuals who rebounded after the antibodies had completely waned. 3BNC117 is a little bit more subtle of a trend, but you see the same association. Our most resistant rebound viruses are able to break through earlier with higher concentrations of antibodies, and then we see relative sensitivity as we get to our late rebound. Okay, so if we go back to this participant 11 and we build from these rebound sequences by looking at the reservoir sequences. Now the reservoir work has been done by Maxime and Matthias in the Raygon Institute, and this is a huge amount of sequencing work that we've done that I'm going to put up here and represent in a very sort of broad sense. But what we have here in black, or here, are the rebound sequences that I just shown you. In addition, they have provided reservoir sequences looking at intact viruses or defective viruses and these large clones that are prevalent in the reservoir. And so I'm not going to be able to provide nearly enough detail to all of the work they've done, but what we were able to do is look at these pre-existing large virus populations, these large clones, and characterize the envelopes from these and compare them with the envelopes from rebound. And so here in blue is actually a clone that was characterized later in the trial, but in vivo we had very poor infectivity and we weren't able to get this to work in our assays. And then our two of our smaller intact clones from baseline we were able to characterize and test the relative phenotype. So here is a highlighter plot looking at our rebound sequences over time. And again, we see just a small number of amino acids accruing over time. And here are our two reservoir clones that are closely related to each other, but actually distinct and quite a bit distinct from the sequences that actually arose in this individual. So what I'm going to show you here is a sort of schema of the trial where we have a period of type 1 interferon induction with antiretrovirals. Then we have our BNAb infusion time period. We can see our levels of BNAbs mapped here, our viral rebound, and the time points that we sampled our sequence. And then I'm going to show you the neutralization sensitivity of plasma from these participants at this time. So at each different time in the trial, the plasma may incorporate different things. So it will have a baseline autologous antibody response. When we've got administered BNAbs, those BNAbs will be part of that plasma compartment. Those BNAbs are going to wane, and then we're going to have the later autologous antibody response that after rebound has responded to whatever antigen was present during that replication and has shifted in its specificities, etc. So that plasma antibody is a shifting and evolving thing over the course of the trial. But in this particular individual, we have our reservoir viruses shown in blue and our rebound viruses shown in pink. And there's not much of a difference between the sensitivity to that baseline autologous plasma antibody in this individual. We have 3 time points, 1 with no intervention and 2 with just the interferon. But we see marginal differences. Now, we don't actually have a sampling during the peak periods of BNAbs, but as the BNAbs are waning, there's still quite a bit left. And we can see a shift in that our rebound viruses are a bit more resistant than our reservoir viruses, which is something we might expect if there's selective pressure from those BNAbs against some of the reservoir viruses, but maybe not those that can break through. And then if we look over time as those BNAbs wane, we see the potency of the plasma come down. And then as our autologous response responds to this viremia and to this new antigen, we see it increase a little bit and sort of get to a new baseline that's a little bit greater, but not substantially greater than our baseline autologous antibody response against both those reservoir and those rebound viruses. So let's look at another participant. This is participant 7. And if we think about this individual, this is another individual who rebounded relatively quickly after those BNAb levels were waning, but after that sort of primary endpoint. So this is in green, and we see a high rebound, a little bit of a dip, and then bounces right back to high viremia. So this person's rebound viruses are quite different than what we saw previously. So here's this first time point at 57 days post the last BNAb infusion. And this is a low diversity lineage. There's actually minimal difference between these two these purple viruses. But when we sample 2 weeks later, and again, this is after this little dip and this come back up, what we see is a totally distinct virus population. So this population has failed to be sampled in that second time point. And what we see here is a new population. This virus is a recombinant between the two, but it's a dramatic example of over just a few weeks, 15 days or so, the pressures in the body allowing one virus to outcompete another. So here we have these 2 rebound viruses, and what you can see at our first time point is that we have a 10-10-74 IC50 that's pretty high. So this virus has got an IC50 of 8, pretty resistant to 10-10-74, and then an IC50 to 3BNC of 0.35, so moderately sensitive. But this virus that outcompetes is very sensitive to 10-10-74, and we can infer that perhaps as that level of 10-10-74 came down, this 10-10-74 virus had a relative advantage for other reasons. The antibody pressure wasn't as substantial, and that may contribute to why we see this rapid replacement with one virus to another. Okay, so in this individual, we can also lean on Maxime and Matthias for some reservoir sequences. And in this individual, there's sampling at 2 periods, one at baseline and one after BNAbs, but before rebound. And if we look at, again, expanded clones from within the reservoir, we take those envelopes and we compare them to our rebound envelopes, we can get a sense of is there differential pressures for our various antibodies. So again, we have this baseline period of time where our first sample has just baseline the study. The second sample is during interferon, still suppressed on antiretrovirals. And again, our 3 reservoir viruses are in sort of blue and green, and our rebound viruses from the first time point and the second are in pink and red. So we can see sort of minimal antibody or autologous antibody differential pressure between these 2 populations at baseline. And then as those antibodies, the BNAbs, are administered, we see a marked increase in the potency of that plasma that has these 2 antibodies at high levels, able to target both the reservoir and the rebound viruses. This comes down a little bit, and over time we can see as viremia is stopped by antiretroviral reinitiation, the autologous antibody response responds, gets bigger or gets larger. It's boosted from what it was at baseline against both reservoir and rebound viruses. So although we see a lot of genetic differences in these sequences between the reservoir and the 2 rebound viruses, we see the antibodies in the host responding to all of these. Okay, last participant, the infamous BEAT-2-006. So again, this individual is shown here partially by these orange triangles. So this individual had viral rebound after the antibodies had waned, and we're only showing part of the viremia in this trial because actually this individual continued to have they rebounded to over 1,000 copies, but then came down and sort of had low-level viremia for quite a period of time, enabling them to be followed as a part of the treatment interruption or the ART restart criteria of this study. So we were able to sample rebound viruses from a long period of time. So we start, and this is probably completely illegible, but we start at day 176 post-BNAPs, and we follow to 392 post-BNAPs. So I'm showing the tree here and the highlighter plot here just to show you, and these first two time points, our purple and our red, we have relatively related viruses that are replicating over the first few months of this treatment interruption. But then over time, we see new distinct populations arise and contribute to this rebound virus. And so we see kind of a varied group of rebound viruses over these many weeks of ongoing low-level viremia in this individual. Okay, so looking at the sequences from Maxime and Matthias, again, this individual has multiple expanded clones within the reservoir, and if they are sort of interspersed between these black sequences down here that are rebound viruses, but we can characterize, again, the phenotypes. So if we start at the very beginning, we see at the first time point, our baseline time point, no intervention. Here are our rebound viruses down here. Here are our reservoir viruses up here. And this suggests that our rebound viruses are quite resistant to the baseline autologous antibody response, whereas our reservoir viruses are much more targeted, much more sensitive to neutralization by the baseline plasma as this participant entered the study. Now, this is an interesting effect, and I don't know how to explain it, and we've checked it about 5 times in our hands, and we will look forward to discussing with others. But when we look at the next 2 time points, when this participant had type 1 interferon in place, we actually will see a marked increase in the sensitivity of these rebound viruses compared to what it was just previously. So we're still working on this, but we have repeated it numbers of times, making sure that we have the right reagents and the right viruses, because it's interesting to me. But if we follow over time, what we can see is at the time point when we have high levels of BNAPs, again, these rebound viruses are very sensitive to the antibodies that this individual received, the 3BNC and the 101074. And you can see these sort of parallel decline curves. As the PK, as the BNAPs wane, so does the sensitivity to these, or the potency of the IgG to these rebound viruses. So we return to this sort of baseline of very substantial resistance to the plasma antibodies at this time, as the BNAPs have waned right before rebound. So as we look over the time points of rebound, what we see is an evolving autologous antibody response. So whereas these 2 viruses were very much resistant to the baseline antibodies, we see those baseline antibodies start to respond after rebound. So we pick up activity against one of these rebound viruses, and then months later, we pick up activity to the second rebound virus. The red is the first, the pink is the second that we've sequentially sequenced over time. And we see a boosting of the potency of the autologous antibody against both of those rebound viruses. And again, it's about the same as the reservoir viruses, which we really don't see a shift. We see that they're well targeted to begin with, and they're really not any different throughout all this time. So the immune response, the B cell response, does not seem to be changing in regard to those reservoir viruses, where it does seem to be making differences to the reactivating viruses that are replicating in this individual, even at a relatively low level over those months of treatment interruption. Okay. So we can put all these things together, and we can think about all of the participants and their relative sensitivity. Rebound viruses, shown here as these yellow circles. Reservoir shown in the yellow circles, the rebound in the open. This is looking at the sensitivity just to the BNAP. So here's 101074. And we can see I've organized these participants in terms of earliest rebound to latest rebound. And you can see that the differential effect, the difference between being sensitive rebound viruses and resistant rebound viruses and sensitive reservoir, it's more pronounced in our early rebounding participants, suggesting that part of the reason why these individuals were able to break through is that those rebound viruses were resistant to those antibodies at baseline or at the time of rebound. And in 101074, I think it's very important to recognize it only takes up one mutation or a small number of reasonably well-tolerated mutations. So it's hard to know when we characterize a virus a few days, a week after replication has started, whether that represents a new mutation or something that was selected from a pre-existing reservoir variant. As we move through time, there's a little bit less of an effect in terms of how pronounced the difference between reservoir and rebound viruses are for 101074. 3B&C is a little bit less pronounced of effect also, but we do see a general trend towards our earlier viruses, our earlier rebound viruses being sort of on the more resistant end of the range of reservoir viruses sampled, and less so when we get to time points with 14 and 16 where the BNAbs have really waned from the system by the time those reactivating viruses start to replicate. So we can look at the same thing with plasma IgG. So this is looking at the baseline plasma IgG. So at the start of the study, these individuals have been suppressed on antiretrovirals. In theory, there's antigen present, but not replicating virus and not a lot of novel virus to stimulate that B cell response. And so if we look at our earliest rebounders compared to our latest rebounders, I think there is, in general, a trend towards our autologous antibodies better targeting our reactivating rebound viruses being resistant to our autologous antibodies compared to our reservoir, but it's definitely more pronounced in our delayed rebounders who have this post-intervention or post-treatment control phenotype, who reactivate long after the antibodies have waned, the BNAbs have waned from the system. So there's many possible reasons for that. When the BNAbs are gone, autologous antibodies may have a greater prominence. Perhaps this PTC, this post-treatment control phenotype, has a whole bunch of other things going on. But anyway, I do think that this sort of phenomenon of delayed rebound and autologous antibody differential pressure against rebound versus reservoir viruses has been shown in this type of analysis, just like it did in the rebound in a dish study preceding me. And so if you look at these three participants over time, just to show the sort of relative difference of the baseline primary endpoint where we have a lot of antibodies and post-treatment, post-study, here our delayed rebounder, there's a substantial difference between the resistance of our rebound versus our reservoir. It's less so with our earlier rebounders. At our primary endpoint, where we have lots of BNAbs in the system still, we see that the reservoir viruses tend to be better targeted, more sensitive to that potent antibody than the reactivating viruses. And at post-trial, we can see in participant 6, we have a substantial boost in that autologous antibody response. In our other participants, it's definitely a less substantial increase in that autologous antibody response. And so just to show that boost over the trial, if we look at the difference between baseline IgG potency and that primary outcome where we know there's lots of BNAb in the system, you can see it's markedly greater against rebound viruses when we have our BNAbs present. But when we look post-trial to baseline, that boosted autologous antibody response is still substantially greater than it was at baseline. When we look at our reservoir viruses, we do see that there's mixed here, but generally there's a statistically greater increase in the potency with BNAbs against the reservoir than at baseline. But when we look at just the autologous antibody response, baseline to after trial, we don't see a statistically significant increase in that response to the reservoir viruses that aren't replicating in any of these individuals. So to sum things up, I would say that combination BNAbs clearly can durably suppress viremia. We saw this as the primary clinical outcome for this trial. And I think from these analysis, we can see that reservoir and rebound virus resistance is clearly associated with those earlier breakthrough viremias. We see that BNAbs can select for or drive new resistance potentially in these individuals, as when we look at the baseline sensitivity measures from the Monogram screening assay compared to these later time points, we see increases in resistance to both antibodies. And whether that's selecting from minor pre-existing resistant variants or driving de novo resistance through the course of this trial is an important question to consider. I think in some participants, our autologous neutralizing antibodies can contribute to suppression or suppressive pressure, and we need to figure out if and when and how that pressure can be leveraged, I think. And so there's a ton of ongoing work has been discussed with all of these different groups to try to put all of the different epigenetic, latency phenotypes, autologous antibody responses, bNAb sensitivity, put it all together and get a more comprehensive picture of the many different factors that contribute to virus suppression in these combo trials. Finally, I'll say thank you to the participants in particular, the study team and the study site, as well as all the collaborators who contributed to this work. And I don't have my acknowledgement slide, but thank you very much.
Questions? Bob?
Okay. A lot of work to process, but I'm struggling with a few concepts. One is, so it seems as if, okay. If I understood you correctly, it seems as if after rebound, there is boosting of autologous neutralizing antibodies to latent reservoir. Is that correct?
There's boosting specifically to the rebound viruses that reactivate and replicate. Sometimes that carries over to the reservoir viruses. Most cases, it seems not to. There's enough difference in the epitopes of those replicating viruses versus the reservoir that we don't see that boost carry over to both. I think over time, we would be interested to see if that boosting does translate to more divergent variants. So there's a lot of heterogeneity in this cohort, and I don't think I did a great job of making generalized themes, but I think that's become from participant to participant, there's really different trends, and it's going to take some time to figure out what the distinct messages are.
So do you think having neutralization capacity of the reservoir is critical to control of rebound viremia in this clinical trial setting?
No. I mean, from the bNAbs, yeah. If you're trying to suppress with bNAbs, then yes, that's absolutely critical. Do I think autologous neutralizing antibody sensitivity is necessary? No. I think it could be complementary to many different approaches, though. It could be a key factor, but I don't think it has to be. I also think in chronic ART initiation, it's really challenging to think about how, at baseline, our autologous antibody response is going to be able to be functional enough and broad and potent enough to basically suppress the diversity of variants in any one individual. But I think in another context, it might be able to be enhanced or improved so that it is a very important feature of suppressing a good chunk of the reservoir.
Thank you.
So Katie, this is probably just terminology, but when you talk about the difference between rebound and reservoir viruses, I mean, the rebound viruses are coming from the reservoir. So it's just that's the subset that is resistant to the autologous neutralizing antibody response. It's basically just another way of saying there's heterogeneity, right, in the reservoir in terms of sensitivity to the autologous.
Yeah, my terminology was clumsy because it's not just the reservoir versus reactivating variants from the reservoir that we're characterizing. It's really the large clones within the reservoir that we're comparing phenotypically and we're able to sample, and not the likely huge numbers of heterogeneous smaller variants. And we're thinking of the viruses that did, for whatever reason, reactivate and potentially shift from what they were in the reservoir, but are closely representing that. So you're right. These things come from one place, and they're differentiated because one reactivated and the others didn't. That may have significance in many different ways, or it may be more stochastic.
A really great talk. Thanks so much for showing all this. Really interesting.
From you.
What I feel is interesting is to look at the fitness of the rebound viruses and so this as an alternative parameter that might be somewhat influenced. It's sort of like the virus able to replicate independently antibodies, and I'm not sure that's something that is in any possible way. And the second question was, you have done this beautiful work with interference. Is it possible to look at?
Yeah, good points. I think that understanding like in vitro and intrinsic replicative fitness is an important thing. And I think the one example where the one virus reactivated and then it was quickly outcompeted by another, maybe intrinsic fitness, or maybe it's T cell sensitivity. I don't know. There's all these different factors. So yes, to do that, it takes even more work than this. We have to make infectious molecular clones, and then we have to have them be able to replicate over time in various primary cells to look at intrinsic fitness, and then in the presence of interferons to look at sensitivity to interferon. So I think those are valuable questions, and we have to think about how we would do those efficiently.
Fine. Joseph, do you want to unmute? Ask a question.
Hey, Katie. This is Joe online. Can you hear me?
Joe?
Hey. So more of a theoretical question, but I'm wondering if you think that a strong neutralizing response, autologous neutralizing response at baseline, is actually related to the ability to generate enhanced neutralization activity after ATI. So maybe there's like a larger antigen-specific memory B cell subset. What do you think about that proposition?
I think that makes a lot of sense. I think if you come from a stronger, more potent, more potentially broad response in that you're targeting many diverse variants of that person's reservoir, you have a better chance of being able to boost that sufficiently to be sort of functionally effective. I don't know that anyone's empirically tested that, but I think that makes logical sense to me.
Thanks. Nice talk.
All right. Go ahead. Okay, 2 more questions.
Sorry, quick question. So very nice talk. So Katie, if you were able to clone the envelope sequences from, say, tissue samples, to put that into your molecular clone, say, would you be to measure the IC50 differences? I wonder, is that because the tissue versus are different?
Yes, we could do that. Oh, that would be interesting. We should put all the groups together with these samples if similar things are being studied, I think.
All right, so we are going to have our first virtual